There are currently almost 6 million people in the United States living with Alzheimer's disease. In the next 40 years, that's expected to triple. In New Mexico, some 41,000 people are living with Alzheimer's. It's a devastating illness that robs people of their mental capacity and brings tremendous burdens for families and caregivers. But researchers at the University of New Mexico have developed a vaccine that carries the tantalizing possibility of preventing this disease. They sat down with NMIF correspondent Megan Kamerick.
I'm joined by Kiran Bhaskar. He's Associate Professor of Molecular Genetics and Microbiology at the University of New Mexico. Bryce Chakarian is Professor and Vice Chair of Molecular Genetics and Microbiology at UNM. And Nicole Maphis is a PhD candidate in Biomedical Sciences in the graduate program. Dr. Bhaskar, I wanted to start with you. You're a longtime researcher in Alzheimer's. How does this disease cause such dramatic cognitive decline? What are the mechanisms that are causing that?
My lab has been working on this disease for almost 7 to 8 years, but my interest in Alzheimer's disease goes back to almost close to 18 years now, since I started my postdoctoral days. And looking at the literature over those many years, and I think we are trying to understand what exactly causes this particular devastating disease and what are the molecular bases and what happens as we age in our brain. I think there's quite a bit of understanding we learned over the years. I think the main culprit that has been thought to drive the neuronal loss in the brain are this amyloid plaque, which is made up of A beta peptide, which is one of the proteins which has a normal function in the brain, but then because it loses its normal function, it starts accumulating in the brain. And
it starts to accumulate.
Yeah, accumulate in the brain. And then there is another protein tau, which is what we are interested in, which accumulate inside the neurons as neurofibrillary tangles. And in addition to that, there is a lot of other things that people have worked on and tried to identify as an etiological factor, like vascular problem in the brain, and inflammation as we age. A lot of stress and things can induce, low grade slow inflammation throughout our, elderly age. So I think those are some of the contributing factors why we see it's going to hit at some of these people who suffer from dementia and lead to brain cell death.
How have researchers tried to tackle Alzheimer's so far, and why is this vaccine that your team and you have found potentially more successful in tackling that?
Right. So yeah, the Alzheimer's research, as I said, is going on for many years. Because Alois Alzheimer, himself described this disease, over 110 years ago in 1907 in Germany, and since then, there was bits and pieces, but past 20-30 years, I would say, it's sort of picked up. And so most of the focus has been on this amyloid plaque that I said, which accumulates in the brain. So people thought that that's the triggering factor which sort of induces all the downstream things, including causing brain cell death. And so that's why people thought that if we somehow intervene right at that stage, we can stop everything.
Stop the plaque from building up.
Yeah, stop the plaque from building, and then if we do that, if we achieve that successfully, we can prevent from brain cells from dying, and also prevent people losing their memory and cognitive impairment. So that was the thought for I would say early 2000s through 2010. And many pharmaceutical companies, they try to target this amyloid plaque with various kind of strategies, and apparently, some of the earliest studies suggested that it might work in some very small population of Alzheimer's cases, but not. It's not like a magic bullet which is going to work on everybody. But then over the years, when they did the follow-up of these particular studies, I think more and more studies, it suggested that I think targeting amyloid is not all that beneficial, as a matter of fact. So that's what sort of led to the researchers to look at some of the other targets, including tau, which we try to generate a vaccination against.
How is the tau different from the amyloid protein?
So amyloid protein accumulates outside the neurons in the brain, but the tau tangles accumulate inside the neuron. So that's the basic difference.
Okay.
And I think more and more studies again suggest that this amyloid accumulations seem to occur as we age. It's like, you can imagine like, wrinkling of the skin or graying of the hair. It may not be after all, the driver of pathology. It just comes up as we age. So that's one of the, debates in the field that targeting amyloid may not be best strategy.
So targeting the tau tangles inside the neurons.
Inside the neurons is once tau also has a normal function. So once tau sort of just loses its normal function, starts accumulating as these fibrils inside neurons, that's when it starts killing the nerve cell.
Dr. Chakerian, I wanted to ask you the vaccine that they've developed. They've used a method that your team has been working on called virus. It uses something called virus-like particles.
Yeah, virus-like particles.
How does it work?
So a virus-like particle is basically, it's a virus particle, okay, but it's not infectious. So it's just like, I think when you think of viruses, often you think of geometric shapes. And so it's just sort of the shell of the virus particle, but doesn't have any DNA or RNA inside of it. And so it can't replicate, it can't infect cells, it's just that protein shell. And one of the things that we tried to do is exploit these structures. It turns out that the immune system has developed all sorts of different mechanisms to respond to viruses. We want to make robust immune responses to virus particles. And so basically we're taking advantage of that property of virus particles. We're taking the shell and then we're displaying stuff on the surface of the virus-like particle. And by doing that, we can take something that doesn't normally induce very strong immune responses and we can make it much more immunogenic.
So the body thinks that it's a virus.
Yeah, basically
and
that's what's
reacts
happening.
To.
Yeah. And so we're essentially tricking the immune system into responding to something. And so we tried taking advantage of this to target all sorts of things that the immune system doesn't normally recognize. And one of these is the tau protein.
So the idea is that it's tricking the body into attacking the tau protein.
Right,
that's
exactly.
built up.
Yeah, and so the way that it works is the way that the normal vaccines that we get works. So you inject it into the body, you make antibody responses against that, and then those antibodies have the ability to basically go anywhere in the body, including the brain, as Kieran and Nikki have shown. And antibodies work by attaching to targets, by clearing the targets or preventing their function, and that's how we think that this vaccine is working.
Is it clear yet if it would work on other forms of dementia? As you mentioned, there are other kinds of dementia, but...
Yeah, absolutely, because I think as tau seem to accumulate as tangles, not just in Alzheimer's, but there are a host of other neurodegenerative conditions that we call it as tauopathies.
Oh, so tau accumulates in
other conditions. For example, this dementia called frontotemporal dementia, where the frontal and temporal region of the brain degenerates specifically. And that's much earlier onset than Alzheimer's disease. So that's where we see this tau pretty much is the trigger of those neurodegeneration. So it could work in those cases. And another good example is the traumatic brain injury, the chronic traumatic encephalopathy, which is which happens following repeated concussions to the brain or acute
There's
brain
tau
injury.
there as well.
Yeah, the tau accumulates in neurons right around this vasculature in the brain, and in those neurons we see accumulation of tau.
Wow.
So we are again trying to test if our vaccine could prevent that, and it could be another option for treatment in TBI.
Nikki, I know it was your task to to test these things, to experiment, testing the
it was
effectiveness of
so
the.
easy.
What did that entail? Mice.
So we
Yes.
use mice in our lab, and the mice mimic the diseases that we see in humans. So we chose an animal model that would present with these, as Kieran mentioned, the tau tangles in the brain. We used an animal model that develops a lot of tau tangles pretty quickly. And we thought, if we're going to test this vaccine, we should try it in the most difficult model. And after injecting our groups, so we set up our groups. So a lot of my task was to set up the entire experiment in terms of the animals we would use and the tests we would use to test whether or not this vaccine worked. And we had really great results.
So you had a control group.
We had our
and then you
control
had-
group when we had our vaccination group. Our control group, as Bryce had mentioned, the virus-like particles, we used a specific type called comprised of Q beta RNA bacteriophage. It's just a different type of virus that we can use to make virus-like particles. And so we had our one group immunized with just the platform alone, and one group immunized with the platform plus our target. And it's the target we chose to pursue is something you see elevated in patients with Alzheimer's disease. Particularly, it's a type of tau that is changed at one specific site, and they use this in biomarkers when they're trying to diagnose. So it's called PT181, and that's what we chose to use. So our vaccine was that peptide displayed on the virus-like particle.
How did you know it was effective?
So we did the entire experiment and injected them. The first thing we did following all the injections, where we did a small animal MRI available at the Center for Brain Recovery and Repair. And I noticed that the animals receiving the vaccine, their brains looked bigger. On the animals receiving the control, they had experienced a lot of brain death. So particular regions like the hippocampus, which is important in learning and memory, and the cortex, which is also important in decision making and learning and memory, were much smaller in the animals receiving the control. So that was my first hint that something was working. And then we did memory tasks. So we challenged the mice to, first of all, remember an object. And this task is called the Novel Object Recognition Task. So the first day I exposed them to two objects, say two of these coffee cups, and the animals will spend equal time exploring. They're naturally exploratory creatures. And then 24 hours later, I would take one of those coffee cups and I would put in a Coke can, for example. Different shape, texture, color, it's in a different place. They should spend more time with that novel object in its novel location. And if they spend more time with the object they've already seen, they can't discern between the two. And so our animals receiving the vaccine spent more time with the novel object. And the animals that received the control didn't really tell the difference. They couldn't see that something was new or changed, or they couldn't remember the day before. And then more excitingly, we did a task called the Morris Water Maze, which is pretty comprehensive task of hippocampal dependent learning and memory. And the animals are tasked to swim through a maze of water, and they find a platform to escape the water. And they use spatial cues on the walls to orient themselves to where this escape platform is located. And we train them over 5 days. The animals receiving the vaccine learn this task much easier than the animals receiving the control. And on the 6th day, after both groups learned the task well enough to find the platform to escape, I take the platform out and I measure how much time they spend. And the animals receiving the vaccine spent more time in the target quadrant. And so we were really excited about that.
So they were looking for the way out because
They found the way out. They remembered
Oh,
exactly
okay.
where it was. They were like, it was right here. Where is it? And they spent all their time right there.
So clearly the initial results say this seems very effective at
Yes,
attacking
yes.
tau and improving brain
Yes.
capability.
But then we wanted to look like
Yeah.
like in the brain. So after all of those experiments were completed, we then took tissue samples from the brain to look at the neurons to make sure that they were intact and they were still functional. And we actually saw that the animals receiving the vaccine had less of these tau tangles. And the animals receiving the control, they were just inundated with all of these tau tangles.
Dr. Chakarian, this seems very promising. So what are the next steps in commercializing this so it can actually get to market?
Yeah, so there's a number of steps that have
Okay,
to be taken, and of course they involve money.
Yes.
And so, there is sort of a process for moving forward with these sorts of vaccines. I think our preliminary data is really, really encouraging for this target, and so I think we're looking for ways to move forward. So this would involve, manufacturing of the vaccine, which is a complicated process, but there have been some similar vaccines that have made it into clinical trials, and so I think there's a really clear pathway for how to move forward. But again, it takes some time, and it takes actually a considerable amount of money to move forward.
So you're looking
with
for
these
investors?
vaccines.
Or-
Well, it's one of the things that we're talking about is, how to commercialize this. One of the things I think that's unfortunate about about our healthcare system and sort of basic science in general is that you really need commercial investment in order to move things from the lab bench into the public sector. And the government helps to an extent. So there are mechanisms where the government is trying to sort of bridge that, that divide. But it only takes you so far. And so, we're exploring a lot of different possibilities for how to move forward.
And Nikki, how has this project influenced what you want to do with your career?
I think it's led me down this really exciting path to explore future jobs as potentially working in the biomedical sector or working as a communicator between biomedical and investors. And so often we have people that work in business or venture capitalists that don't necessarily understand the impact of research, and so they may invest in flashy things, but they're missing the stuff that could really mean something. And so, potentially a job in venture capital firms would be a nice way to kind of lead me away from the bench, but I do love biomedical science.
I think it would be she's really good, and I think it would be a big loss to lose her research. But,
yeah. But I do love the business aspect of science, and I think that there is unique opportunities there.
So it sounds like this will take some time, could be years before it comes to market, but we would welcome you to come back and talk about how it's progressing.
That's great.
That'd be great, yeah.
Thank you so much for joining us.
Thank you.